Good
Good
day.
day. I am Beverly May Constantino,
I'm JV Galan,
and
and
this
this
is
is
the
the
isolation
isolation
of
of
DNA
DNA
from
from
cheek
cheek
cells.
cells.
Before we discuss the isolation of DNA from our very own cheek cells, let us first recall what DNA is. Deoxyribonucleic acid, or DNA, is the molecule that relays genetic information for all organisms. It is mainly composed of deoxyribose and phosphate groups, as well as four nucleobases: adenine, thymine, guanine, and cytosine. Due to the uniform composition of DNA amongst living things, isolation techniques and reagents are almost the same for each organism.
Now let us isolate DNA from cheek cells. For this activity, DNA isolation will be done using household items. Cheek cells were extracted by gargling saline solution for 30 seconds and recovering it with a plastic container. Concentrated dishwashing soap was then added and stirred into the solution. An equal amount of ice cold isopropyl alcohol was then added into the cup and left for 3 minutes. DNA filaments were then observed and food coloring was added to further visualize the isolated DNA.
After the conduct of the procedure, three main observations could be taken from the isolated DNA from cheek cells. First is the appearance of the DNA isolate, second is its low yield, and third is its inability to hold onto the stick upon isolation. The DNA isolate appears to be cloudy in the solution and has a slimy texture. The yield of DNA from the extracted cheek cells was low, as you can see here in the pictures. Additionally, the DNA isolated from the sample does not firmly hold onto each other, as when they are attempted to be extracted, the strands would fall off the stick and then would break into smaller fragments.
Now let us find out why the isolated DNA exhibited the characteristics mentioned by Bev. The first observation is the appearance of the DNA isolate itself. DNA isolate from the solution was found to be slimy in texture. Along with this, it appears as cloudy strings and or clumps in the solution upon precipitation. This string-like appearance can be explained by the structure of DNA itself. DNA is a very long molecule composed of nucleobases, deoxyribose, and a phosphate group. In small amounts, DNA cannot be seen without the help of laboratory equipment. However, with high enough amounts of DNA present in the sample, one may see DNA with the naked eye, as such with this activity.
The second observation is the low yield of the isolate upon precipitation. The low yield of DNA from the sample may be explained by the limited amount of cheek cells harvested from the investigator. Since DNA is found in cells of living organisms, the number of cells present in the sample greatly affects the amount of the isolate. If cheek cells are abundant in the sample, it would result in a higher yield. However, since the method used for harvesting cheek cells in this procedure involved the scraping of the teeth against the cheeks and gargling saline solution for 30 seconds, it is expected that the number of cheek cells will be minimal.
The third observation is the inability of the DNA strands to cling onto the stick when it was attempted to be extracted from the solution. Along with this, the DNA strands and clumps would be broken into smaller pieces after contact with the extracting stick. Now, this may be explained by forces involved during the isolation of the DNA from the sample, such as the stirring force after the addition of detergent. Forces like this are able to shear DNA into smaller fragments, resulting in its inability to stick to one another during the attempted extraction.
The relevance of these results mainly concern the retrieval of cells to be used in the DNA extraction. If one aims to have a high DNA yield from a sample, it is highly recommended that DNA extraction be done at a larger scale and with a better source or a better cell harvest. The more cells present in the sample, the higher the DNA yield is going to be. Along with this, the temperature of reagents, especially the alcohol to be used in order to precipitate the DNA, contributes to the amount of isolate that can be retrieved. Colder temperatures facilitate the higher yield of DNA upon precipitation due to the inhibition of enzymes that destroy DNA that is present in cells. These results confirm that DNA is indeed a long strand of molecule and can be easily isolated from available cells in a sufficient amount.
There are three main steps done in the isolation of DNA from cheek cells: cell harvest, cell lysis, and DNA precipitation. In order to harvest cells in the activity, a saline solution was used. In this step, the reagent used was salt with a chemical formula NaCl. Since DNA is negatively charged due to the phosphate groups in the DNA backbone, the sodium ions neutralize the negative charge of DNA by interacting with the phosphate groups. This does not affect the harvest of cheek cells, but is important for the precipitation of DNA since the interaction of sodium ions and the negatively charged phosphate groups of DNA makes the molecule less hydrophilic due to the neutralization of charges.
In cell lysis, the cell is disintegrated, releasing the DNA it contains. In this step, the reagent used was concentrated liquid detergent. Liquid detergent can dissolve lipids and proteins that hold the cell membrane and the nuclei together. This is attributed to the polar head and nonpolar tail of the detergent, wherein fats are attracted to the nonpolar tail of the substance, while the polar head is responsible for the solubility of the soap lipid complex in water.
In DNA precipitation, the added salt in the solution during the earlier part of the activity serves its purpose in this step. Along with salt, a reagent used for DNA precipitation was ice cold isopropyl alcohol. DNA is soluble in water, but it is insoluble in alcohol. This is due to the difference in the dielectric constant of water and alcohol, along with the interaction that alcohol makes with water. Alcohol has a lower dielectric constant than water, which is critical for the successful interaction of the sodium ions to the phosphate group of DNA, as alcohol protects this complex.
When interactions between the sodium ions and the phosphate groups are successful, this decreases the hydrophilicity of DNA. Additionally, when alcohol interacts with water, they form hydrogen bonds. The formation of such bonds allows the water molecules to interact more with the alcohol than with the DNA molecule. Because of this, the DNA is extracted from the solution into the alcohol.
Next, we will be discussing four qualitative tests that you may use in order to determine the presence of DNA in your sample. These qualitative tests include the Schiff's test, test for deoxyribose, test for purines, and the test for phosphate, each of which have different ways of detecting DNA.
The first qualitative test for the presence of DNA is the Schiff's test. Schiff's test is specific for aldehydes, and here DNA must first undergo hydrolysis in order to test positive. The reagent used here is Schiff's reagent, which contains fusion dye, acid, and bisulfite. In this test, bisulfite binds to the central carbon of the fusion dye in the presence of acid, resulting to a decolorization of the dye and a colorless solution. However, upon addition of aldehyde from the hydrolyzed DNA, a pink solution is formed, as you can see here in the picture. So, the positive result in this test is a pink solution, which means that an aldehyde is present. A negative result in this test is a colorless solution, which means that an aldehyde is absent.
The test for deoxyribose, or the DISH diphenylamine test, is specific for deoxyribose only and will not work for ribose. Therefore, this test can be used to differentiate the presence of DNA from RNA. DNA must undergo hydrolysis first in order to have a free deoxyribose molecule, and the reagent used in this test is the diphenylamine reagent. In the presence of an acid and with heating, the reagent forms a blue colored complex with the deoxyribose in the sample, resulting in a blue solution. The intensity of the blue coloration depends on the concentration of DNA in the solution. A blue solution would mean a positive result for the test, and a colorless solution would mean otherwise.
The next test is the test for purines. In this test, hydrolyzation of DNA is not required. Along with this test can give positive results for both hydrolyzed and unhydrolyzed DNA. The reagents used here are ammonium hydroxide and silver nitrate. Silver nitrate interacts with purines through the interaction of silver ions with the lone pairs of the nitrogens in purines. Hydrolysis of glycosidic bonds between the purines and the deoxyribose or ribose in the presence of ammonium hydroxide results in the release of adenine and guanine. So the positive result for this test is the formation of white precipitate in the solution.
The last qualitative test that we could use is the test for phosphate. In this test, the sample must be incinerated first to get rid of other elements such as carbon, nitrogen, hydrogen, and oxygen from the DNA. Elements exit the sample in the form of gases, and only phosphate will be left behind. The reagents used for this test are ammonium molybdate and concentrated HNO3. Upon the interaction of ammonium molybdate with the phosphates, it produces a yellow complex. Now, the formation of the yellow precipitate indicates a positive result for this test.
After the isolation of DNA from cheek cells, can we consider the extracted DNA to be pure DNA? Well, the answer is no. Other substances may still be present in the extract, such as protein and cell fragments, since the activity only covers isolation of DNA from cheek cells, not purification of it. Additionally, low molecular weight substances present in the solution may also act as a contaminant in the DNA isolate. These substances may include salts as well as detergents. If one chooses to obtain pure DNA from the extract, one may subject the isolate into various purification techniques in order to get rid of the contaminants.
Before we end, let us have a quick recall of what we have learned. First is that DNA is the molecule that relays genetic information for all organisms. Through the DNA isolation from cheek cells, we had a glimpse of what it looks like through the naked eye. However, the isolated DNA does not purely contain DNA. Along with this, the conduct of the activity has helped us understand how different substances work in order to isolate DNA from a sample. Lastly, there are four qualitative tests that may be done in order to determine the presence of DNA: the Schiff's test, test for deoxyribose, test for purines, and the test for phosphate.
And that concludes our presentation. Once again, I am Beverly May Constantino.
And I am JV Galan. Thank you for your time, and I hope that you've learned something from our presentation. Peace out.